Hi everyone, my name is Jason, and today we're going to learn about investigation design, one of the most important things in any scientific experiment. So to start off, what is investigation design? In essence, it is a framework used to design a specific scientific experiment. It lists out a step-by-step process that all scientists should follow and details ideas that scientists should consider along the way. For instance, if you take a look at the graphic on the right, there's a step-by-step process where we follow through by posing a research question, a hypothesis, an experiment, and from there we can draw some conclusions. And so all scientists should follow the same investigation design so that they can properly answer the question they pose and so that other researchers can repeat experiments in their own labs. So the very first step along the way is posing a question. Every scientific experiment known to mankind starts with a question. What happens when you mix water and oil? How do humans perceive colors? How is the universe formed? All of these are questions that were first posed by scientists that have now been answered today by various scientific experiments. By formulating a question, scientists can take the first step to designing an experiment that will find an answer. So in today's scenario, we're going to talk about a researcher who wants to learn more about daphnias, small aquatic animals that live in freshwater. And if you have no idea what a daphnia looks like I did, here's a quick image of it on the right-hand side here. And they live in freshwater environments like huge lakes or ponds. So the researcher would like to know how different water temperatures affect the heart rate of daphnias. So how would you pose this curiosity as a question? It's a lot more simple than you might think, actually. I would just take the second bullet point that we just saw, maybe cut off the first part of it, and just rephrase it as a question. How do different water temperatures affect the heart rate of daphnias? And there we have it. We have our question for the day. So what we have to move on to now is a hypothesis. A hypothesis is a prediction. You are thinking about a possible answer to your question before you actually do the experiment, and they are often stated in an if-then format. So for instance, our hypothesis could be, if the temperature of the water goes up, then the heart rate of daphnias will also go up. Now, note that this is a very important point. Your hypothesis does not have to be correct. You are predicting something, but it doesn't necessarily have to be a correct prediction. You're just sort of framing your question in a way so that you have an idea of a possible answer, but your conclusion is going to determine whether or not your hypothesis was correct or not. Now, we're going to actually design the experiment that we're going to run. So the very first question you have to ask is, how will you run the experiment? For the purposes of today, let's say that we're going to measure the heart rate of daphnias before and after you change the water temperature they swim in. For instance, let's say our initial temperature is 23 degrees Celsius. That's going to be our base rate or our baseline. Next, we're going to measure the heart rate of daphnias after we switch them into a different water environment, let's say 4 degrees Celsius. We're then going to determine the change in the heart rate to determine how the change in temperature has changed the heart rate of the daphnias. So what are we going to change throughout the experiment? Like we mentioned earlier in our hypothesis and in our question, we want to know how the temperature of the water changes the heart rate. That means that throughout the experiment, we're going to be changing only the temperature of the water. And so consequently, a question might be, what are you going to keep the same throughout the experiment? Well, if we want to know the exclusive effect that temperature has on heart rate, we need to keep everything else the same. And that includes the amount of water added, the type of organism, and the initial temperature of the water, which we already defined as 23 degrees Celsius. And when we're designing this experiment, we can't leave out this conversation about variables. So there are two different types of variables. There's an independent variable, which is the variable changed by the researcher. In this scenario, the only variable that the researcher changes is the temperature of the water. That's the only thing that's going to be in our control. The dependent variable, on the other hand, is the variable that changes because of the changes in the independent variable. In this scenario, the dependent variable is the heart rate of the daphnias because we're not actually changing the heart rate, we're changing the temperature. But we're predicting that the change in the temperature is going to change the heart rate of the daphnias. The important point here is that there can only be one independent variable, and that's because if we change too many things around, we're not going to know what causes the change in the end. For instance, if we change both the temperature of the water and the volume of the water and saw a decrease in the heart rate, we're not going to know if it was the temperature change or if it was the water volume change that really caused the change in heart rate. That's why in every scientific experiment, it's really important that you only change one thing at a time. Now, here are some general rules about doing the experiment. Now, the first thing that you want to keep in mind is that you want to repeat the experiment multiple times. And that's because this increases the accuracy of the data because taking an average reduces error in the measurements. For instance, in one trial, you might mess up really badly. But if you have multiple trials that you did perform correctly and you took an average, your data is going to be more accurate or representative of what it was intended to be. The second point that I want to make is that it's very important to have a control. You want to make sure that you take a measurement of the heart rate without changing the independent variable. This is to prove that the heart rate will not change unless there is a change in the temperature of the water. Otherwise, if the heart rate changes when the temperature stays the same, that indicates that something else might be going on, and then we don't want to use that Daphnia for experiment. The last point I want to make is the difference between observations and inferences. So an observation is when we gather information using our sources. And this is when we're doing something like counting heartbeats. We're using our senses to visibly count the heartbeats. An inference, on the other hand, is when we make a statement about observations using additional knowledge. For instance, we know that heartbeats are increasing because of reduced oxygen in the water. Now, I said we know, but really we don't know for sure because we can't see the reduced oxygen in the water. And so this is an inference that we're making, or almost kind of a guess or a conclusion that we're making based off of some of the little contextual cues that we gather across the experiment and outside of it as well. So now that we've actually done our experiment, let's take a look at some example results. So here we have the change in heart rate in a nice table laid out. In the top row, we have the different temperatures, 4 degrees Celsius, 15 degrees Celsius, 23 degrees Celsius, and 37 degrees Celsius. On the left-hand corner, not in the left-hand corner, sorry, the left-hand column, we have the different trials. So as you can see, we ran 3 different trials for each temperature. At the very bottom, we have our averages. So let's focus on the very bottom row for now, the average row. We have at 4 degrees Celsius, there's a change of negative 26. At 15 degrees Celsius, there's a change of negative 20. At 23 degrees Celsius, which by the way is our initial temperature, there's a change of 1 degree. And at 37 degrees Celsius, there's a change of 37 degrees Celsius. So as we can see, as the temperature seems to go up, our change in heart rate seems to positively increase. Whereas if we go in the other direction, the heart rate actually decreases. So what conclusions can we make from here? Well, to start off, to define what a conclusion is, a conclusion determines if the initial hypothesis was correct or not. So based on the results, we see that as temperature goes up, the heart rate of the Daphnias also goes up. On the other hand, as temperature goes down, the heart rate of the Daphnias goes down as well. Thus, let's go back to our original hypothesis. Our original hypothesis was that if the temperature does go up, the heart rate will go up as well. So the findings of this experiment support our hypothesis, and we have answered our question. And there you have it. So to quickly recap, the investigation design is pretty much the scientific method, or a step-by-step process that you need to follow for every experiment that you conduct. Outlined here is a quick graphic that just summarizes everything we talked about. So the basic points: first, we need to pose a question that we need to answer. Next, we need to predict the outcomes of the problem by formulating a hypothesis. Then we develop a procedure to test the hypothesis using an experiment. We run the experiment to actually collect the data. We analyze the data and then create a conclusion that is later compared to the hypothesis to determine what our experimental results were. So I hope that was helpful and you learned a lot about investigation design.